Hello, everybody. Thank you for joining us in this webinar. Today, we're going to have a discussion about understanding natural history studies, and the speaker is Dr. Alfred Paul. I want to go over some webinar reminders first before we introduce Dr. Paul. First of all, the purpose of this webinar is to provide general information and is not intended to replace the advice of your or your child's doctor. Dr. Paul and CCF will not be able to provide any recommendations regarding your particular child or family member, or comment on their specific treatment. If you are enrolled in the MYPBC3 Natural History Study and have specific questions, please contact your study site, coordinator or principal investigator for information. The webinar will be recorded and available online after the event. As usual, we will leave 15 minutes at the end for questions. So throughout the webinar, make sure that you submit your questions in the Q&A box and use the chat box to share general comments and report any technical issues that you may be having. With that, I want to introduce Dr. Alfred Paul. Dr. Paul is Professor Emerita of Cardiology at Northwestern Feinberg School of Medicine and at the Ann and Robert H. Lurie Children's Hospital in Chicago. As a clinical scientist, Dr. Paul has spent her career working to improve outcomes for children with cardiomyopathy and heart failure. Dr. Paul has collaborated with the North American Pediatric Cardiomyopathy Registry on multi-center studies focused on genotyping and biomarks, and led the genetic study on children with hypertrophic cardiomyopathy. Dr. Paul is also serving as the chair of the Pediatric Heart Transplant Study Foundation. Dr. Paul, the floor is yours.
Thank you. Next slide. Good afternoon, patients, parents, and caregivers of children with cardiomyopathy. Next slide. I'd like to thank Tanaya and the Children's Cardiomyopathy Foundation for the opportunity to speak to you today. And I would also like to disclose that I am a consultant for Tanaya Therapeutics, and Tanaya Therapeutics prepared these slides for this presentation. Slide. So what is a natural history study? This is a medical research study which is very important and performed generally in patients who have a rare disease or medical condition, such as pediatric cardiomyopathy. A natural history study describes the course of the disease over time, both with and without standard treatments that are available. And natural history studies improve the understanding of the condition for us today. Next slide. Natural history studies give us a pathway to have a better understanding of rare diseases such as cardiomyopathy and gain a big picture view of what happens to a patient over time when they have this diagnosis. These studies can also help improve understanding of the rare condition by looking at individual people's experiences with the disease. Information that's collected includes demographics, such as which populations of people are most affected, which age range are affected, is this a disease that occurs in families and has a genetic component, and in which parts of the world do we see the highest numbers of patients with this disease. Natural history studies will also look at the disease itself in terms of severity for that patient based on their symptoms and on laboratory and imaging criteria such as echo or MRI. We would also be interested in genetic information if it is available, to know if the disease occurs in families, and to get an idea of the sense of number of hospitalizations that that patient has undergone and undergoes going forward, and also any events that occur in the future, such as need for surgery or devices. Another way to look at the burden of the disease is for the families, as well as the patient, to fill out quality of life questionnaires, which are short surveys that give us a better sense of the impact of the disease on the patient and their family. Natural history studies may also identify new patterns of disease that can only be identified when we study large groups of patients. Next slide. There are two primary types of natural history studies. Retrospective studies only look backwards and are looking at what has happened to the patient up until the time of enrollment, and often is a one-time collection of information from the medical records. In some cases, a retrospective study may also look at patients who have died from a rare disease or patients who have undergone heart transplantation. On the other hand, a prospective study is very exciting as it looks at individual patients when they are enrolled and going forward over time as the disease progresses, and can be several years long. A prospective study will collect new information from the medical records, often performed at the time when patients have a routine clinic visit, and will give information on that condition over several years. This information can also be collected for patients from their past medical records, and it'll be important to have that background baseline information for all prospective patients as well. Next slide. So there are several reasons why we do natural history studies. These studies give us powerful information and conclusions about a particular rare disease, because we're able to study large groups of patients over many years, often from numerous medical centers. And they matter because it helps understand the natural course of a disease over time and will help with the development of new therapies. These studies provide essential scientific foundations for clinical developmental programs of pharmaceutical companies. And they also enable us to compare new treatments to the old standard treatments to look for differences. Drug developers can therefore design more efficient and relevant clinical studies. Next slide. As I mentioned, the most robust natural history studies are those that are done in large groups of patients over time and include multiple medical centers. They examine the current medical treatment for these patients and can uncover opportunities to meet gaps in our current therapy and help drug companies determine what is needed to help care for these patients with a rare condition. Next slide. In pediatric cardiomyopathy, we have patients who have a rare disease of the heart muscle, which affects the ability of the heart to function well. Pediatric cardiomyopathy affects 1 in 100,000 children in the United States from studies that have been performed by the Pediatric Cardiomyopathy Registry. The photo on the panel shows a specimen from a child who underwent a heart transplant and had a very severe form of hypertrophic cardiomyopathy with thickened heart muscle shown in the middle as well as the sides of the lower chambers of the heart. We know that there are multiple types of cardiomyopathies, with dilated cardiomyopathy being the most common. Dilated cardiomyopathy is a type of heart muscle condition that causes the heart chambers to swell, enlarge, and thin out and stretch and function poorly. Hypertrophic cardiomyopathy, the second most common type of cardiomyopathy, is a condition where the heart muscle becomes unusually thick. And in adults, this occurs in 1 to 500 patients, but maybe even 1 in 500 adults, but maybe even more common than that. Rarer types of cardiomyopathies, such as left ventricular non-compaction and restrictive cardiomyopathy, also exist in children. LVNC, or left ventricular non-compaction, is a rare condition of the heart muscle that results from abnormal prenatal development and can be diagnosed in infancy or later into adulthood. Restrictive cardiomyopathy is very rare and is a condition where the chambers of the heart muscle become very stiff over time with large upper chambers and very poor function for the patient. Next slide. This cartoon is a genetic depiction of a portion of the cardiac muscle, and it highlights some of the genes that can be abnormal in hypertrophic cardiomyopathy. For instance, The incidence of the gene change for an abnormal gene in hypertrophic cardiomyopathy for MYBPC3 is approximately 20%, probably the most common gene abnormality we find for hypertrophic cardiomyopathy, both in adults and in children. Individuals with changes in the MYBPC3 gene can also have other conditions such as LV&C or dilated cardiomyopathy. And this is especially the case for the very young patients, the infants who have inherited 2 copies of the abnormal gene, usually 1 from each parent, and they have the most severe form of cardiomyopathy. Genotyping or gene testing and clinical screening with imaging of all family members when a patient has cardiomyopathy is recommended by the American Heart Association guidelines from 2020 for hypertrophic cardiomyopathy. And in 2021, a new guidelines paper specific to children with heart conditions recommends that all children with cardiomyopathy undergo gene testing. Next slide. For pediatric hypertrophic cardiomyopathy, there is a dire need for new therapies and ideally cures. Treatments are highly individualized and depend on the specific cause of the cardiomyopathy as well as other risk factors. The chest X-ray shows a patient with HCM who underwent internal cardiac defibrillator for primary prevention of poor events. Risk assessment includes an assessment for risk for sudden death, arrhythmias, as well as extreme left ventricular muscle obstruction. There are some medications that can provide relief of symptoms, with the most common being beta blockers, and less common in children are calcium channel blockers, as well as a few new drugs which have not yet been tested in children. Some surgical therapies may be beneficial, especially for a patient who has extreme narrowing of the left ventricular outflow tract, and heart transplant may be the only option if all of the treatments fail. Currently, there are no medications that cure any type of cardiomyopathy, but there is hope for potential investigational treatments that may provide better treatments and even hope for a cure beyond our existing medications and therapies. Next slide. In order to develop new treatments for hypertrophic cardiomyopathy in children, this is an extreme challenge because it's a rare disease and we have small numbers of children who have this disease. Often there's a delay in diagnosis due to lack of education or understanding among a medical professional who may be seeing the patient with unusual symptoms. And often patients don't present until they have severe progression of their disease. We have a very limited understanding of the natural history of cardiomyopathy in children, as well as its progression over time, because there have not been many studies on this in the past. And we have no good understanding of what the best treatments and practices are for caring for children with cardiomyopathy. As far as drug companies developing new therapies, there are no prior drug development pathways that we can readily follow for cardiomyopathy in children. And we don't have any FDA guidelines to follow as well. Clinical trial outcomes, what a study hopes to achieve, must be relevant for that patient and their particular disease, and must also be approved by the Federal Drug Administration, FDA. Next slide. So to emphasize our current situation, when we're speaking of pediatric cardiomyopathy, we have scattered information from a few researchers on small numbers of patients. And some of these studies are very interesting and important, but we have little information available on how this rare disease progresses over time in children. Thus, doctors and patients can often find themselves unprepared to anticipate what's next and to manage such a rare disease. A natural history study can provide future directions, both for treatments and to look importantly for the gaps in treatment that currently exist. Next slide. I'd like to discuss next the TN-201 program for MYBPC3 cardiomyopathy, which is Tanaya's clinical development program. Next slide. We'll speak first about gene causes of hypertrophic cardiomyopathy. Genetic causes have been identified in about half of adults with hypertrophic cardiomyopathy, and we expect it to be similar in children. Genes provide heart muscle cells with instruction to produce proteins to have healthy heart contraction. The incidence of an abnormal MYBPC3 gene in children and adults with hypertrophic cardiomyopathy is approximately 20%, or 1/5 of patients with HCM. Changes in the MYBPC3 gene are commonly identified, and a non-working gene can lead to decreased MYBPC3, which is a critical protein in the heart muscle, and thus non-functioning of the components of the heart muscle or the sarcomere. And this ultimately leads to the left ventricular hypertrophy or thickening and the picture of hypertrophic cardiomyopathy. MYBPC3 gene therapy is currently under development. Next slide. This slide highlights some of the components of the TN-201 program, which includes the natural history study in children seen on the bottom panel. And this has already begun last year, and we're continuing to collect additional centers to participate. This was necessary for our pre-IND work, with IND being an investigational new drug application in conversations with the FDA, who recommended that a natural history study be performed. In addition, Tanaya has initiated a phase 1 study application for adults with hypertrophic cardiomyopathy or severe type of MYBPC3 cardiomyopathy caused by, similar to hypertrophic cardiomyopathy. In this study, a very small number of adults with severe disease will be tested with gene therapy. And this initial pilot study will look at safety and to see if the gene therapy actually makes a difference. If this study goes well, then a phase 1 infant study will be performed next in the critically ill infants who have 2 copies of the abnormal gene. And last, the larger phase 2 and phase 3 studies would be done in children and adults to further test the gene therapy before a biologic license application, a BLA, can be undertaken. Next slide. So we'll talk a little bit about the natural history study, which is called the MYCLIMB study. Next slide. The MYCLIMB study is a pediatric natural history study for children who have cardiomyopathy caused by an abnormal MYBPC3 gene. Most of these patients will have hypertrophic cardiomyopathy, but we would also be interested in studying patients with other forms of cardiomyopathy caused by changes in the MYBPC3 gene. The objective or goal of the study will be to look at the outcomes, risk factors, the severity of illness, the quality of life, and also blood tests to look at biomarker changes in these patients who have cardiomyopathy. It is a hope that these results would then inform the development of a clinical trial design of new therapies, including gene therapies for children with this condition. Next slide. If your child is enrolled in the MYCLIMB study, we would obtain health information from the medical records. And in addition, the patient and caregiver would be asked to complete a survey about their quality of life. In addition, blood would be collected at the time of routine visits for biomarker testing if the families are interested and consent. And in addition, if there is any surgery that occurs and there is tissue available from that surgery, we would also collect the surgery if we have consent. Next slide. Currently, the study began in 2021, and we anticipate that it'll end in 2028, so that the majority of patients will have had 5 years of follow-up if they are in the prospective study. We have enrolled several sites in North America, as well as the United Kingdom and in Europe, and are continuing enrollment of additional sites. As I mentioned, there are 2 categories of patients that participate in the study. In the retrospective cohort, we expect to enroll 100 patients who have changes in the MYBPC3 gene, looking only at past data that is available. And this could include a child who had undergone a heart transplant or had passed away. In the prospective cohort, we will be looking at 100 individuals who are enrolled and will continue to be followed over a 5-year period with information that is collected at the time of their routine clinical visits with their medical provider. For entry into the prospective cohort, patients must meet certain criteria, such as symptoms or specific findings on their cardiac imaging, such as echo. We'll be enrolling children from infancy through age 18 and are particularly interested in finding young infants with severe disease who have changes in both MYBPC3 genes and have the most severe type of cardiomyopathy. Next slide. If you are interested in participating in the MYCLIMB study, please look at the website myclimbnhs.com or speak with your healthcare provider. Next slide. In summary, natural history studies give us a path to have a better understanding of rare conditions such as pediatric cardiomyopathy. There are 2 primary types of natural history studies: past retrospective studies, which only look backward in time, and prospective studies, which enroll a patient and follow them going forward over a matter of years. Natural history studies will help us streamline the study design to help provide important information so that we can improve the treatment and care of children with cardiomyopathy. TN-201 is Tanaya Therapeutics MYBPC3 cardiomyopathy clinical development program, which includes components that are the natural history study as well as therapy. The new natural history study in pediatric MYBPC3 cardiomyopathy sponsored by Tanaya is the MYCLIMB study. And last, gene therapy for cardiomyopathy is under clinical development. Next slide. We also have 2 patient advocacy representatives who we would welcome you reaching out to if you have any further questions about the MYCLIMB study: Wendy Borsari and Lori Ann Correa, who can be emailed at patientadvocacy@tanayathera.com. Next slide. And now we'd like to take some questions.
Thank you so much for that very interesting and informative presentation, Dr. Paul. We have a couple of questions that came in throughout the presentation. The first one being, do all patients with cardiomyopathy have a change or mutated gene?
This is a great question because we continue to learn about genetics and the development of diseases. And the frequency does depend on the type of cardiomyopathy. So patients with hypertrophic cardiomyopathy have the highest percentage of a likelihood of having an abnormal gene. It's about 50 to 60%. However, if you're a patient or a child who has dilated cardiomyopathy, it's a bit lower, it's about 30%. And in some individuals, we may not find an abnormal gene when we do gene testing for a certain number of genes. We know which genes we're looking for now, but that disease may still occur in families. And so gene testing alone is not enough. It would be important for the families to also get clinical screening. And new genes are regularly found. And so the gene list is updated when we look for genes that cause cardiomyopathy. A long answer to a short question.
Thank you very much. The next question that we have is, why should families with cardiomyopathy get gene testing?
So there's a concept called cascade screening, and this concept relates to the fact that we may identify one family member with cardiomyopathy. It could be a child, it could be a parent, and because so many cardiomyopathies do run in families and have an abnormal gene component, it's important to test going up to the parents or grandparents, test the siblings, and then test the children of an affected patient with cardiomyopathy. Patients could have a gene mutation and have no symptoms at all and not be aware that they are at risk for developing cardiomyopathy, and thus would benefit from risk assessment and routine medical follow-up. And on the positive side, if genetic cardiomyopathy runs in the family and we know which gene has caused it in the affected individuals, a child may be screened and not have the gene, and this could free them up from significant further follow-up and additional worry for that family. There are also guidelines from the American Heart Association, and these are similar from the American College of Cardiology and the European societies that recommend gene testing for all family members of a patient with hypertrophic cardiomyopathy. And importantly, last year, there was also a recommendation for gene testing in all children who have cardiomyopathy.
Great. I'm just looking at the chat right now, and I just want to make sure that everybody knows that the email that was shared in this slide was wrong. The correct one is patient.advocacy@Tanaya Health. TanayaEra.com. So if you can just make note of that, and the correct one is in the chat, so you can just copy that and keep it as reference in the future. The next question that we have is, how many patients will be in the natural history study, and when will we see results if my child enrolls?
We hope to recruit approximately 100 patients in the retrospective study and 100 patients in the prospective study. And because the prospective study will go forward for at least 5 years, probably up until 2028, it'll be some time before we see the final results. It's possible that Tanaya may decide to perform an interim analysis when they've enrolled about half the population.
Okay. The next question is, will gene therapy cure cardiomyopathy?
That is a great question, and we don't know. We don't know yet at this point. There are important animal models for different types of genes that are affected in cardiomyopathy, and we are hoping to investigate gene therapy in people, in humans, in the near future, and then we may be able to give a better answer.
Okay. The last question that we have is, why is this study only looking at the MYBPC3 gene and not the MYH7 or other common mutations?
That's an excellent question. MYH7 is a much larger gene and more difficult to deliver. And so Tanaya has decided to focus on MYBPC3. The two of them are the most common forms and causes of hypertrophic cardiomyopathy. And the FDA, when they were approached by Tanaya, had mandated that Tanaya do a natural history study specifically on patients who have MYBPC3 cardiomyopathy to get more information about the disease progression and disease course on the affected patients. And then the key reason that MYBPC3 was chosen for this natural history study is that is the particular gene for which there is a very exciting animal model that Tanaya has with encouraging results on improvement in cardiomyopathy.
We actually had one more question come up, and it's, does the genetic therapy in the MYCLIMB study use stem cells?
So the MYCLIMB study is not offering any type of therapy. The MYCLIMB study is a natural history study where we are looking at what happens to a patient who has hypertrophic cardiomyopathy. The gene therapy that is being considered in the future and currently is an animal model is not with stem cells.
Okay. Well, we don't have any other questions right now, but I do want to say thank you very much for this webinar. The information that you gave us was great, and we will be posting the recording of this webinar in a few days. Look for the email reminding you of the recording, and we'll also share it with those that couldn't make it here. But thank you, Dr. Paul. Thank you for your knowledge and sharing that information, and we wish everybody a good night.